Okay, sorry for this. We are just trying to figure out the hybrid is always still difficult. But in the meantime, I will introduce Thomas Hartung. He is the Dernkamp-Sinton Chair for the Evidence-Based Toxicology with a joint appointment for Molecular Microbiology and Immunology at Johns Hopkins Bloomberg School of Public Health. And he holds a joint appointment as Professor for Pharmacology and Toxicology at the University of Constance in Germany. And Thomas is the director of both Centers for Alternatives to Animal Testing at Johns Hopkins University and at University of Constance. There we have the European division. And he's the former head of the European Commission Center for the Validation of Alternative Methods, ECWAM, and he has authored by now countless scientific publications. And I will hand it over to him now.
So good morning, everyone. It's an honor and privilege to be here. Usually you're doing your meetings at a time point when I'm spending my summer in Italy. This year, it was a bit earlier, so it worked out, and I'm very happy to be part of this. I would have loved to see more people live and in person. And when we will have to move forward then, I'm sharing my slides. I put an QR code here, but also on the last slide again, and Katrin will certainly be happy to bring this forward. I've been given the task to talk about the orphan 3Rs. Eric Hutchinson heard me talk about this earlier. It is well known that the replacement, reduction, refinement have fathers. Russell and Birch shown here, actually Birch and Russells in the sequence they are standing here. They fathered in 1959 the concept when there was not really alternatives around. We neither had cell culture nor computer methods which could have possibly considered as an alternative to animal testing. So it was really a visionary approach. And this visionary approach took until the 70s, and it was slowly rediscovered and then became now a mainstream concept for anybody involved in the life sciences. I think essentially all animal welfare legislations, but also the different scientific organizations have embraced this concept as a vision. Some years ago, we were musing about the idea of adding additional Rs. Can you go forward, Anwen? And these, let's say, orphan 3Rs, which have no father, or at least were not fathered by Russell and Birch, and I could not name anybody who precisely has fathered them, which we coined at the time, was read across, which I will show to you as a concept which is mainly getting new life, or getting at the moment on speed, I could say, by the availability of artificial intelligence and an automated type of read across. The next R, which I think is especially relevant, is relevance, because I think we have to think about aspects of reproducibility, both of the animal and of its alternatives. It is about good practices. It is about how we handle information properly, and it is about reporting standards on both sides of the aisle. And then the last R, roadmaps. It is about how to bring things into action, how to actually proceed, how to manage change. And this is a very, very multifaceted aspect. It is about communication. It's about the conferences, the policy programs, the stakeholder engagement, a lot of things where our Center for Alternatives to Animal Testing here in Hopkins for 42 years almost has worked as an information hub, trying to help, I often call it the lubricant in a big machinery. We are not more than a humble academic institution, but very often with the flexibility this gives, we can help that certain movements, certain changes are taking place a little bit faster. Next, please. I have to alert you of a couple of conflicts of interest. I don't see any of them really as critical interests, because I'm not selling any products to anyone, but I'm trying to help to get alternative approaches implemented. Please take everything which I'm saying with a grain of salt. Next. Our work is also sponsored by a variety of companies. You can construe this as a conflict of interest as well. But 3 times more money next comes from philanthropic sources, which gives us a certain independence. Still, our work with industry is critical, because if we do something which is the industry worth paying for, we do something which is actually having an impact. And next, we do actually receive most of the funding for our research part, because it's very quickly to burn money in the laboratory. We receive from the normal competitive resources of the European Commission, the FDA, the EPA, the Department of Defense, and the NIH. So with this said, if you miss the logo of your company, if you have a rich uncle or whatever, next, I always find space on this diagram. As you can see, I left the space open here in the middle. Next, Anwen. Yeah, you see, that's the space reserved for you guys. Okay, next, please. We all know that there is a lot of movement in this area, and we have seen over the last 20 years about 10–15% increase in the Americans objecting to medical testing with animals. About split half, as so many things here in the US, the population is by now. These numbers go up to 70% if you ask for animal testing for cosmetic purposes. And they are on average about 10% higher if you look into Europe. And this public opinion shapes politicians and their views. And we should be clear that society expects us to act on this. And this has led to legislation in Europe. I don't need to repeat this, on chemicals and cosmetics, but also the US has more recently been taking up these concepts, implemented 3Rs in legislative things like the TSCA reauthorization. We heard in 2019 that the last administration wanted to phase out animals as a standard requirement by the EPA by 2035. They have somewhat taken back this commitment to move out of animal testing by 2035, but the EPA continues major efforts to reduce animal testing and, for example, with annual conferences, a roadmap, and many individual activities. The European Food Safety Authority has announced they want to get out of animal testing as a standard requirement by 2027. The European Medical Agency has announced they want to implement major measures. And we all have seen that the FDA Modernization Act, signed by Biden on the 23rd of December, urges the FDA to take into account non-animal methodologies. So I think the playground is clear. The public and the policymakers want serious changes in this field. Next, please. Our center is trying to serve this triangle here of science, public, and policy, which has many players, from NGOs and their advocacy programs, for the media, the different lobbyists which are working in this field. We are trying to, as I said, try to be the lubricant. And we have a little bit expanded on this in a recent article in my Food for Thought series. Again, the QR code is here. That's how we define ourselves. We want to help that there is a win-win-win situation between the various partners involved in this game. Next. So let's talk quickly, just very quickly, about the 3Rs, the classical 3Rs, before I expand on number 4, 5, and 6, I would suggest. Replacement is obviously the ultimate goal. We would hope to replace animal testing wherever possible. And I think there is one very big game changer which we have to notice, and this conference series obviously has addressed this several times. While especially human cells came with a lot of handicaps and quality and quantity available, we had to rely mainly on tumor cell lines, which are certainly not reflective of the organ. Otherwise, we could implant a tumor into a recipient organ as an organ for an organ recipient. So it's a but the advent of stem cells only in 2006, in really ethically acceptable way as induced pluripotent stem cells, has paved the way for bioengineering of what is really advanced cell culture, which is reflecting organ architecture and organ functionality. That's what we call microphysiological systems. Next, please. They come in many flavors. I don't want to go into these details. You can essentially open any journal in the life science and you will see a new protocol, a new device or something coming out. All organs can somehow be cultured. Sometimes not yet with full maturation, but the protocols are getting better and better. Next, please. This is my early time, my claim to fame, perhaps. I put this up because in 1995, I developed a pyrogen test. So a test for the identification of microbiological contaminants in infusible drugs. I developed a whole blood pyrogen test, so something which is based on human immune reaction. I put this up because already a year later at the second World Conference for Alternatives, after the first one was created by our center here 3 years earlier, Bill Russell was actually in the audience when I received the award. That's a younger version of myself here to the upper left from Prince Laurent from Belgium, who is sitting also next to Bill Russell here, down here. I bring this up because I want to show you the difficulty in replacing an animal test. This essay, as I said, developed in '95, and there's a few sister essays, which I will mention in the next slide, please, Anwen. These essays are a field of essays which we validated jointly. I put together all of the groups who had proposed validation, alternative methods for the rapid pyrogen test based on the human blood reactions, which are now known as monocyte activation tests. And this is history, yeah? 2002 to 2003, I was the PI for the validation study with 10 partners, which included some national control laboratories, the European Pharmacopoeia, and the European Center for the Validation of Alternative Methods. And probably this has shaped my life because afterwards I was offered to become head of the European Center for Validation of Alternative Methods, because it was one of the early successful validation studies which showed that an alternative method can actually outperform an animal test. And with this, we moved forward. And next slide, please. We invented this in 1995. In 1995, a kit version of this was made commercially available, so very quickly. Validated 2006, took us roughly 10 years to run this through a validation study and the formal acceptance. It was accepted then by the FDA 3 years later, by the European Pharmacopoeia 4 years later, and by the ISO in 2018. And the US Pharmacopoeia has a type of mentioning of it, but not really a full implementation since 2017. Shows you how long this path is. Next. I summarized some of this in a paper when we've celebrated the 25th anniversary of the whole blood monocyte activation test. So this is available. I'm wondering why the QR code is missing here. Okay, sorry for this. Next. But it took even some years longer to really get animal numbers down. We have good statistics as usual only from Europe. This is a paper which did address this. Next. What you can see here now is how the number of rabbits did decrease in Europe for pyrogen testing. In 2005, so around the time when this assay was declared valid, we were at 160,000 rabbits. It even increased a little bit towards 2008. And only now you see how this is dramatically falling. Actually, the slide is somehow cut short. The 2020 data, which were released just 2 months ago, they show now 20,000 rabbits. So down from 170,000 rabbits here to just 20,000 rabbits. You have to compare these numbers next. In 2005, as the point of comparison, there was only roughly 100,000 animals each for entire chemical safety testing and about 100,000 animals each for pesticide evaluation in Europe. So rabbit pyrogen testing still was a major consumer of animals. And the very infamous eye and skin tests with rabbits used about 5,000 rabbits per year in comparison to 170,000 rabbits for pyrogen testing. Yeah. Shows you that this was really a major contribution. And next, the big news in Europe was in 2021 that Pharmaeuropa, which means the European Pharmacopoeia, will phase out rabbit testing completely within 5 years. So another 3 years to wait until they will eliminate the monograph for rabbit pyrogen testing. It will be no longer possible to use rabbit pyrogen testing in Europe. We have no such implementation in the US, unfortunately, but this is a different story. Okay, shows you 30 years to get something rather simple, which is in many aspects superior to the animal test, has positive, negative controls, is quantifiable, and whatever name it, it shows the human reaction, not the rabbit reaction. It is less disturbed than other assays, but this group of assays is really took 30 years to get to something here. Okay, next, please. If you had told me this when I was a young postdoc inventing it, I would not have pursued this work, I would assume. Okay, lessons learned. That's why I put it up. Alternative methods can be better than animal tests. They are really enabling technologies. For example, we can test pyrogens on solid materials, which no other assay can do. Very important for medical devices. I think the success of these assays was because they were mechanism-based and using human cells, reflecting the human fever reaction. I've learned about the importance of commercialization to make them available, but also to see that sales forces of companies are bringing this into laboratories to really make use of these. 15 years from development to acceptance, 30 years to full implementation in Europe, not worldwide. I mean, Brazil has accepted it, some others did, but it's really a very, very long ride to get this done because we lack enforcement. While the law is reasonably clear, if there's an alternative reasonably available, there's still nobody telling people there's something reasonably available and please change. It was, I think, a success story because it established large public-private partnerships between technology providers, users, and agencies. And it was critical that we had international harmonization. Still, the fact that Japan, for example, felt that this assay is valid, but they don't need it, when they evaluated it in 2007, is a major hurdle for any company which is trying to sell globally these type of drugs. We're continuing to work on this. I'm in preparation of a workshop towards animal-free pyrogen testing here in the US, but it is a difficult road. For example, I applied last year for becoming a member of the US Pharmacopoeia working group for microbiological safety, which is responsible for this and was not accepted for this. Though I think, it's all trying to stay humble, but I think I'm still one of the foremost experts about pyrogens and especially non-endotoxin pyrogens, but they didn't want to have this voice on their panel. Yeah. Okay, next slide, please. The field of replacement is moving, as I said, towards human cells and microphysiological systems. I wanted just to alert you that we were able to initiate a series of world summits for microphysiological systems. And we organized last year in June in New Orleans, the first of these world summits. You see here Don Ingber from Harvard and Susie Fitzpatrick from the FDA as my two co-hosts for the first meeting. I'm the PI next for a total of 52 organizations which teamed up in New Orleans. And this was a hybrid event. Of the 665 registered participants, 65 came from the FDA, showing you the enormous interest in this new technology by the FDA. And you cannot underestimate the importance of this. The FDA is the agency in the world which sets the standards for pharmaceutical industry. And pharmaceutical industry has the deep pockets to further any technology and really get it implemented. And why it's the FDA? We are 4% of the world population here, but we are 48% of the drug pharma market. 48% of all money for drugs is spent in the US, and 64% of all money for drugs under patent. So pharmaceutical companies only look for what the FDA wants. This is why the FDA Modernization Act, which is only highly symbolic, it is not really changing anything legally, is so important. By the position of the FDA to further these technologies, they even twice funded our research on microphysiological systems, and they funded many other projects, which is very unusual for the FDA, who's otherwise only on the receiving side. We bring the conference now next to Berlin next month. 26th to 30th of June will be the second of these, and next. The progress is enormous. You see the hosts of this meeting. We have now, in the meantime, about 60 organizations. We have more than 1,200 registered participants, so we are tripling the attendance of people. And we will I'm looking forward to an exciting meeting end of June. Next, please. Critical for all of this work is quality, because if we don't quality assure these things, they will fail, and a failure of such tests in safety sciences and elsewhere will be dramatic. So we have been pursuing the quality of cell models, good cell culture practice, and published last year, next, good cell culture practice, which was an initiative of mine already starting in the 90s and published in 2005 by the European Commission. We have taken this up again and expanded this now to these microphysiological systems and human primary cells commercially available and others. So there is some guidance available on quality assurance, and we are working on reporting standards and trying to help the field to exchange information in high quality. Next, please. With this, I would like to quickly mention our own research, which is motivated by autism. This is why we are into microphysiological systems. 2 months ago, the CDC released the current data on autism. You see it now is 1 in 36 children born in the US showing being diagnosed with autism by the age of 8, up to from 1 in 44 just 3 years earlier. So it's an enormous increase, which is, as you see, 400% since 2000, which you can no longer attribute to better diagnostic and awareness because the standards have not really changed in this period of time. So there is something happening here. And we have been asking us next whether there is a contribution of chemicals. Is there developmental neurotoxicity involved from early on? Next, teaming up with Alan Goldberg already in 2005, we addressed the animal test, which was the only option at the time. One of the most expensive animal tests which there is, the protocol by EPA or OECD, is about $1.4 million per chemical, is using more than 1,000 animals, and it is not working. An analysis of the EPA of the last 200 chemicals tested clearly showed there was no regulatory consequence of spending $300 million. So it's an assay which is simply not identifying what we need to identify. So next, in 2005, we teamed up with my old shop and my current shop, which is Equam and CAT. And we said we have to do something about this and develop, in the complete absence of any alternative, we developed a strategy quickly joined by the EPA and later by the European Food Safety Authority and OECD. We developed concepts, a community. With 9 workshops and 4 international conferences, we shaped a group of people, which is now largely overlapping with the expert group on developmental neurotoxicity of the OECD. And the great news is they have found agreement on a guidance document, which at the moment undergoes some blessing procedures and will be published later this year on how to do developmental neurotoxicity testing in vitro. And we will have next year the 5th of our international conferences to be organized by our center in Constance, Germany, the European branch of CAT. Next, please. Our own work led to brain organoids, a microphysiological system. Lena is leading this area here with us. So we developed in 2016 standardized human 3D brain organoids from induced pluripotent stem cells. You see one next here, beautiful brain organoid. These are round balls of brain equivalents which are electrophysiologically active, show all the different types of neurons and helper cells. So it's really a very interesting and versatile model. I can be very short here. Next, if you just click through. We have done this from different genetic backgrounds. We have studied different diseases, various infections, cancer grafts in this model, toxicity, obviously. And we're doing a lot of work towards functional assays of these. Most notably, most recently, we are embarking on organ intelligence, and I will say a word or two on this in a sec. Please, next. You see here the autism rates in the US, how they're increasing once again. And if you click again, if you see, there's astonishingly an uptake now, which raises some questions. Is this really an actual increase which is taking place? Next, which could be linked to the pandemic, which we're just experiencing. Why so? Next, we asked ourselves very early on whether brain cells could be infected with the coronavirus, with COVID-19. And already in May of 2020, we were able as the first worldwide to show with our brain organoids that this neuron here is infected with SARS-CoV-2. Each of these red dots is a virus particle. Next, the Financial Times and 200 others reported on the time about our findings. We published this in June after peer review. And next, there's also a paper a year later showing that 10 other groups have repeated our findings and that they're in line with clinical findings that a certain proportion of people shows virus infection of the brain. And it could very well be that this is impacting also on autism rates, as inflammation and infection is one of the risk factors to develop autism. I'm not saying this is, but it is something to look at. It is showing how an alternative method is an enabling technology readily available if something hits. We still have no animal model which is showing the neurological participation or consequences of COVID-19 infections. Next, please. Okay, I mentioned organoid intelligence. This is one of our latest things which I have to share with you. We are trying to combine the organoid with a computer, in essence. We're asking what happens since they are electrophysiologically active, what happens if we give them input and output and feedback on their output? So we really want them to learn. And there's increasing evidence that these type of systems are capable of doing so. Our partners in Cortical Labs in Australia published in November that brain cell cultures can play the computer game Pong. They're now working with our brain organoids because we have also the machinery for long-term learning to really get something imprinted. We have the glial cells necessary for this, and we are working with various partners on toxicology and drug development projects, creating models of Alzheimer-derived patient cells and autistic children, and trying to find whether we see cognitive deficits by the ability of these systems to perform very simple learning tasks. We got next funding from Hopkins, different types. This is, for example, a type of EEG which we developed. These are electrodes which are embracing our brain organoids and allow us to measure the electrophysiology, which is occurring spontaneously, and then in response to communication with the outside. Next. We published 28th of February this vision paper on organoid intelligence, the new frontier. This came with a lot of editorials and political statements. Next, the kit version and the lay people version. We published a workshop report and so on. There's a lot, and this was taken up so far in about 600 press hits, made it to all of the major press worldwide. It is a concept which is inspiring. The White House took this up as their bold biotechnology goals, engineering, something like this. And we succeeded to get the National Science Foundation to now launch a program on engineering organoid intelligence. So we hope that this is forming a community and doing something. Next. So I could not resist to talk about, about replacement, which is simply closest to my heart. But let's talk quickly about others. I think at the moment, the biggest potential for reduction of animals is in the combination of various test types. I think that we have essentially no overpowered studies anymore. Nobody is using unnecessary many animals. They're simply too expensive. The non-invasive methodologies sure have some potential, but I think here is really a lot of music by in vitrosi, as I like to call it, in vivo, in vitro, and in silico combined testing strategies which place the animal test only at the very end, where a lot of decisions are taken before. We know this now as integrated approaches to testing and assessment, or defined approaches like the OECD is calling this. If you're interested, this is last year's summary on where we are with these developments. Next, please. Yeah, refinement, I feel almost uncomfortable to talk to you, mainly invested into animal experimentation. I did my share of animal experiments early on in my career, but I'm certainly not a specialist in this field. Next, but for us, it is really important that refinement is one of the 3 R's we want to work on, as we see this as an ethical responsibility and good veterinary and scientific practice. But we also are very clear, we don't see refinement as a substitute for possible reduction and replacement. It is only what we have to do until we can reduce or refine or replace. Next, and I was fortunate to find partners in crime, first with Jan, who is here with us, and then with Katrin. And these are 2 of their papers which described what we can do as an academic center with very limited resources. And this is still what is guiding us in the limited work we can do. Okay, next, please. Now, this is still something to alert you. There's a number of classes which are taught by Katrin, which we are at the moment offering for the Hopkins environment, but which we want to bring now to Coursera, so an open online platform, because we have seen that there is an enormous interest in such academic offers. Next, because the first 2 classes which we brought to Hopkins, Toxicology of the 21st Century and Evidence-Based Toxicology, they have now more than 17,000 active learners enrolled over the last 4 years. Yeah. So you see that from very small classes, which we have here, for a lot of money, these free offers are taken up worldwide, and there's an enormous interest, and many people are writing us, thanking us about what the materials we are providing. One of my short-term goals is to now make some of these classes, as well as some of the classes in green toxicology and computational toxicology, available through the same pathway. Next, please. The next orphan R is read across, as I said. Read across is something which came up in the literature around the year 2000, and is a concept of, do I really need to test if I know enough about similar compounds, or can I infer that this substance here in the middle, which has not been tested, is also an active if everything which is similar is active? That's called read across. It is a data gap filling, concluding from structurally similar chemicals. It is something which is scientifically perhaps questionable, because we have seen in the case of thalidomide that even the enantiomers have completely different effects, but it is working, because in the end structure is very often leading to effect. And next, it is something which has been broadly used, especially in the context of the European REACH registration, because the legislation is wide open for it. It calls for it. We have seen that in the first 2 rounds of registration, which are really the expensive and heavily used chemicals, 75% of all registrations used read across. While we did not see that REACH, despite the fact that I pride myself for getting the promotion of alternative methods as one of the 3 goals of REACH into the legislative context, they're not used. Because essentially, it's marginal what has been used in vitro and other in silico methodologies. Read across is wonderful. Expertise in the industry, however, is low, because there was no guidance at the time available how to do read across properly. And as a consequence, ECHA only accepted about 25%. In all of the other cases, sent the people back and said, do the animal experiment. Next. So we started a program on read across, asking ourselves, next, go the 4, top. Is this perhaps the fourth R for the chemical industry with this possible use? While the pharmaceutical industry mainly refines the animal use, pesticides try to reduce by integrated testing strategies, and cosmetics have to replace. Is this perhaps the right R for chemicals? So next, what we saw is that there was need for good practices. So we brought 35 people together and developed jointly what we call good read-across practice, GRAP. And we also discussed at the time, what can we do beyond chemical similarity towards biological data, biological similarity. Next, this is history as this took place in 2016, but it now becomes highly relevant with the advent of artificial intelligence. I mean, nobody who reads a newspaper will not hear about how artificial intelligence is booming at the moment. And the reason for this is that we have a synergy of data, computing power, and AI models. Over the last years, the data in the world are increasing by about 60% every year. Or you can say it a different way, in the last 2-3 years, about 90% of all data in the world have been generated. This is incredible. At the same time, our computers are increasing in power, Moore's law, for the last 50-60 years by twice every second year, or you can say 40% increase every year, is the computational power, unbroken. Yeah? But AI is doubling its potential every 3 months for the last 10-12 years since deep learning was introduced. So 8-fold increase in 1 year. Next. And this is making it a disruptive technology. And over the last 60 years, since computers were introduced, there's about a 1 billion-fold increase in power. Our iPhones already 10 years ago were essentially as powerful as the computer available to land on the moon. Yeah. This is just, it is mind-blowing, yeah, what is taking place here. And now we tried, what can we do for read across by using AI? Next, please. And this is a paper we published in 2018, where we did exactly this. Next, I will be short as this is a bit historic. But we built a large model for the 9 most commonly used animal tests for toxicity testing. Together, they are consuming 57% of all animals in toxicology in the EU. 57% of toxicology. And we showed in the end that this model was capable for 190,000 chemicals, where we had a hazard classification to correctly predict this in 87% of the cases, which compares favorably to only 81% reproducibility of the respective animal tests. So it is showing you an enormous potential shown for 190,000 chemicals. And we predicted each and every one. We did not allow the program to say, I'm not certain, we sort this out, because that's the nicest way to boost predictive capacities. So the work was covered twice in Science and twice in Nature. So you see there was enormous interest in this approach. Next. And we have been extending this as animal AI-based replacement in the meantime in a number of studies. Just to quickly mention them to give you a flavor of what is happening since. Next point, we were interested, how well does this predict human data? So we took human skin sensitization as an example where we have human data. And for 560 chemicals, we predicted them 80% correctly, while the local lymph node assay was only capable of 74% identification. And the local lymph node assay is at the moment the preferred method. Last November, we published a study where we showed we can do this en masse. We took a database of 4,700 chemicals, which are food related. So they're either added to food in the US or they are direct contact materials. And we did run the equivalent of 38,000 animal studies, which would cost more than $250 million in 1 hour. And a small subset was used for evaluating how well we predicted. And we were 83% in alignment with the animal studies which were available for this. Yeah. So it is possible to run essentially a chemical universe if you only run your machines a bit longer. And we just presented for the first time as a poster at SUT, the Society of Toxicology, in March, that also systemic toxicities can be addressed. We predicted 75% correctly cancer risk of 950 REACH-registered chemicals, and 82% correctly the reproductive toxicity risk of 1100 chemicals. If you see that the reproducibility of a cancer bioassay is only 57%, yeah, this is really an enormous advance, and not even 1% of the chemicals on the market in developed countries has been tested in a cancer bioassay, because it costs $1 million, takes 5 years. This is done in minutes, yeah, and might help to just say, this is something to look for problems. I'm not saying we can decide on the basis of these tests, but it is integrating a lot of information to guide whatever we want to do. So I see a lot of prospect for read across, especially AI-enabled read across for the future. Next, please. And this is showing you that at the moment we work in a larger EU project, a consortium which received for 18 partners $20 million. We are addressing liver, kidney, and developing brain using exactly these methodologies, and I have the privilege to lead the AI part of this project. So it is just doing this on now for systemic toxicities. Next, please. We also think about how to use these things not only for regulatory toxicity testing, but something which is available from a structure based by pressing a button can also be used by chemists. They can evaluate, is a structure even worth synthesizing if it comes with possible liabilities. This idea we called green toxicology, and since 2014, Alex Mertens in my team is leading these efforts. Next. This editorial in Toxicological Sciences, Green Toxicology, Know Early About and Avoid Toxic Product Liabilities, is describing what the idea is about. We are trying to educate chemists, educate product developers that this type of evaluations can lead you to early on say, let's take another substance which is probably better. And Alex next has published last year the first textbook as a single author textbook on green toxicology, which is really laying out these concepts. Next. This brings me to relevance as the fifth of the R's and second orphan R. Next. Relevance is what all is about. But a point which has not been properly addressed from my point of view is, what is the relevance of the animal model? And I'm a toxicologist mainly. Toxicology should actually have the best animal models possible. They're highly standardized as OECD test guidelines. They've performed under good laboratory practice, a quality assurance scheme by skilled performers, not the young students. We have high doses. We don't need disease models. These are healthy animals. And so we just study strong effects. So next, please. This is a slide I owe to my friend Nicole Kleinsteuer, as heading the US validation body. And you see her summary of some of the assays, Eutotrophic and Hirschberger, even OECD validated assays. They show the reproducibility of these in large scale studies. And my conclusion of this is the value of animal studies is typically very much overestimated, and they give us difficulties as a point of comparison. I cannot validate something and show that my tests are better than 85%, 90%, what typically is expected from a validation study, if the reproducibility of these assays is not there. Next shows you our data from the already mentioned study. Just to give you a flavor, we really had for the six most used animal tests, we had 350 to 750 chemicals which were tested at least 3 times. All OECD guideline studies under GLP. Some were done extremely often. We found 2 chemicals which were tested more than 95 times in rabbit eyes. Yeah. Just to give you a feeling about the data set. some 100 times repeated. This was in total 2,800 chemicals for which these multiple tests were available. And the reproducibility of these animal tests was on average 81%. But this was inflated by the negative substances, because the negative substance stays negative. The positives were only 69% reproducible, which means that running the same animal test 3 times leads to a 2-to-1 result. And if you do it only once, you have 1/3 probability of missing the toxic property. This alone should open up very much for complementing these animal studies with other approaches or replacing them if you have something better. Next. Anwen? Yeah. Okay, just to be clear, cell cultures are not per se better. They have very similar quality problems. And we are very happy if one of these models is also relevant. I've published largely about this, but I just want to raise the flag. It is not that anything is better just because it is alternative, is different. Ethically, perhaps, yeah. But we have to assume that about 25% of the cell lines used in academia are misidentified. They're often not the species we are thinking, it's not the organ we are thinking, not the sex we are thinking. There are studies continuously over the decades, and this has not really dramatically changed. We have to assume that 15 to 25% of academic research is done with mycoplasma infected cells, yeah, which is a dramatic impact. The genetic instability of these cells is crazy. We analyzed MCF7 cells, a breast cancer cell line from the 1970s. More than 30,000 scientific papers have been done with this cell line. But it lacks, for example, 10% of all its genome. 10% have been lost. About 30% of the genome was, however, multiplied up to 32 alleles of the same gene we found. And 50% was there with less than 2 copies. So it is really a completely reshuffled genome, and we should not expect anything. We did a follow-up study 2 years ago, which showed they do not even reflect breast cancer. They have no similarity genetically with breast cancer, where they originally derived from, because they were cultured too long. Yeah? So you cannot even sell them as a good model for this. Okay, next. I also think that relevance is not only ethical and scientific, but it's also economic. This is an article we published in 2018. The most important omics is economics, and we simply try to describe what are the economical environments in which this is taking place. And interestingly, I was very blown away that Markets and Markets in their independent assessments came to the conclusion that toxicology, which we see always as an animal science, is about 4.2 billion per year in vivo, but is 14.4 billion in vitro work. So there is a dramatic change in the use of these. I found also very interesting this figure. In the EU, as I said, we have good statistics, and we could see that in only 6 years, from 2005 to 2011, pharma industry dropped animal use by 40% while increasing their research budgets. And so they are clearly moving out of this. And we also can show that our capacities, our throughput is simply not enough for the new things. In 2014 already, there were more than 7,000 flavors in e-cigarettes, none of them toxicologically evaluated. Good luck to implement this. So there is a discussion to be had about what is the economical view. And then suddenly ethics becomes an aspect of marketing and corporate culture. Safety has something to do with the liability and defendability in court. Science is about product development, and regulation is about trade policy and barriers for trade. We are only scratching on the surface, but it is actually one of my most downloaded papers. 12,000 times this has been downloaded, showing you the enormous interest in this. Next, please. This brings me to the last of the orphan Rs, which is the actionable part, the roadmaps. This is about the education, the communication, the workshops, the strategic plans, and how to handle the information better. Next, I want to highlight only a few aspects here. First, it is important to understand toxicology, which is the one we are mostly addressing in our work. Next, is actually only consuming about 10% of animals, but I would say it's fair to say that 90% of work on alternatives is being done in this field, which means a lot. Means also that we can expand a lot of these concepts now to other areas of the life sciences. Next, please. Yeah, that there is such a need. I mentioned already that we are a bit working with outdated methodologies. And this is how technology advances. And now next, here you can see when the different animal tests were introduced. The cancer bioassay in 1970 is actually still one of the youngest. This is also now more than 50 years old. This is the pillars of regulatory toxicology. Everywhere else, things are changing. Next. The landscape in toxicology has changed dramatically with the National Academy of Science report in 2007, Toxicity Testing in the 21st Century. However, the knowledge in the life sciences is doubling. There's different estimates. 7 years is the most conservative. 3 to 5 years, have others said. The next is doubling so fast that since 2007, we have now at least 4 times more knowledge than we had at this time. So AI is not mentioned. Microphysiological systems are not mentioned because stem cells were developed in 2006, deep learning in 2010. So this could not be anticipated. So it is time next for version 2.0 on how to implement and change in our field. And that's exactly what we try to attempt. Next. In about a week's time, this report will be published. A future directions workshop by the Department of Defense, advancing the next scientific revolution in toxicology. They approached me 3 years ago, and I found 2 co-chairs and 30 experts who joined us last year in Arlington. And with 15 agency observers, we developed jointly a report, which will be published by the end of this week or early next week. Next, please. The workshop report is shown here. It is building on the workshop on the National Academy of Science report, is just saying, what can we do better now? So this is a roadmap for changing toxicology. Ultimately, it is calling for a human exposome project, similar to the Human Genome Project. And this is an article which I published already in January, because I, at the time, understood our report would be published also in January. So it is a bit already of a preview available on this concept of a human exposome project. Next, please. So I think that science at the moment is transforming. And this transformation is occurring away from the traditional thing, one experiment at the time. I have some beliefs, some experience, some observations, and then I run experiments for causation and proof. That's the traditional way of hypothesis-driven research. There's 2 major changes at the moment taking place. Next. One is that we are looking into machine learning and AI on the right, and use big data and derive the evidence by machine learning, which is at the moment really changing science a lot. I have not the time to discuss this, though I'm happy to do this. I'm talking a lot about the topic. But the other one is applying evidence-based approaches, which means to distill the most relevant data and to use this piece of evidence. So it's something very different. The best humans can do, evidence-based approaches, systematic reviews, and risk of bias analysis and whatever, or machine learning as a machine-driven approach. Next, and this idea of evidence-based approaches in toxicology is something we have been pursuing since our publication here in 2006 towards an evidence-based toxicology, at the time still at ECVAM in Europe. We held next the first conference in 2007 for evidence-based toxicology. And from this resulted next the chair for evidence-based toxicology I'm holding here in Hopkins. And next, since 2011, we are having the evidence-based toxicology collaboration. So this is really a movement of handling the information in the most objective and transparent way, not asking about saving animals. It is not about any of this. It is just saying, how can we undisputably evaluate methodologies and come to conclusions from the multitude of studies we typically have available. Next, and over the last 12 years, you see the enormous success this has had. From essentially no systematic reviews in the environmental health sciences, we are now approaching enormous numbers. More than 1,000 people engaged with our evidence-based toxicology collaboration. We just founded the first EBT journal with Taylor and Francis, and we are now converting the evidence-based toxicology collaboration, which was run by a board of trustees, which included FDA, EPA, European Food Safety Authority, and others, to a membership-driven organization. So we are at the moment expanding this and changing gears here quite a bit. Next. One of the big implementation paths forward for us is the problem that we have to move from black and white to shades of gray. One of the big problems in toxicology is that we always want to know this is toxic, this is non-toxic, and just expect our methods to deliver this, while science actually is full of uncertainties. There's a lot of grays, and every result comes with some uncertainties. As soon as we embrace this, I think we come to a situation, next please. Thanks, Jared, for saying no worries. I hope nobody has to worry. Okay, this is moving us to concepts of what we call probabilistic risk assessment, where we no longer say this is carcinogenic, this is not carcinogenic, but everything is a probability of an effect with some uncertainty around it. And I believe this will be very important for the integration of new and alternative methods, because if we accept that each and every method is just contributing to more certainty, and it is much easier to integrate the new with the old. And we are developing these concepts also in the context of OnTox, and we are doing workshops on how to do exactly this, so that in the end, we come to a probability of risk and an information economics where we also can see which method is contributing how much to our knowledge and to our evaluations. Very exciting work, which is at the moment in the beginning, but already receiving enormous interest by the OECD test guideline national coordinators, which had me present on this. And so it is really now with AI something which is enhanced dramatically. Okay, next. So I think I gave you some flavors here of how the field is really expanding from the traditional 3 R's of replacing, reduction, and refinement to something which is embracing other technologies, things which are no longer these simple categories, because we are integrating our knowledge. It is a lot about no longer about the availability and development of these methods, but about change management, about handling this information in an objective way so that we actually can observe change. And John Maynard Keynes, an economist, as you know, said very nicely, the difficulty lies not in the new ideas, but in escaping from the old ones. And I think this is really the important part. So for us, it is important to be part of this change of ideas. And this is what our center is meant for. Thank you very much for having me here, and I hope you have 2 wonderful days.